Hello, my name is Aun Zaidi, and this presentation is about the blend segregation study using lab automation. This is the research which I carried out when I was a co-op in GlaxoSmithKline in their high throughput automation lab. My advisors for this project were Eva Wu and Parin Desai. So in this presentation, I'm going to take you through each one of this section. So talking more about the background of segregation. What exactly is segregation? Segregation is basically the demixing, or it is the opposite of mixing. Mixing is when components in a blend gets homogeneously distributed, whereas segregation is the opposite of mixing and it will affect the blend uniformity because the particles or the components in the mixtures will not be homogeneously distributed, and you will find certain regions in the mixture where the concentration of a particular particle or the particular component is more than another region where the particle is or the component is lower in concentration. So you will have a very non-homogeneous blend, and that is when we will say that segregation has happened. So in the pharmaceutical context, segregation can pose some very serious health consequences. Segregation in a pharmaceutical setting can occur in different places. It can occur during the fluidized bed drying, during milling, blending, or even during tableting. So imagine if you're trying to make different tablets from a big blend. You want all the tablets, all the small tablets to have a uniform concentration or uniform components of all the mixtures so that it reflects what was in the big blend. But if segregation has happened, what will happen is that uh, the components or the concentration of components from tablet to tablet is going to vary. So what that will mean is that you're giving potentially wrong dosage um, to the customers, and that can pose some very serious health consequences to the patients. So what exactly causes segregation? There are several mechanisms that can come in play and cause segregation. I'm going to take you through a couple of them. A detailed description on each one of them is beyond the scope of my presentation today. So one mechanism that causes segregation is percolation. What happens during percolation is that size difference causes the finer particles to sift through the coarser ones. What I mean to say by that is, if you take, for example, this picture in which we have the blue particles and the brown particles. The blue particle is much larger in size as compared to the brown particles. So what the brown particles can basically do is they can over time sift through the spaces between the large particles, and over time what you will see is that the brown particles have a higher concentration in the at the bottom, whereas the blue particles have a higher concentration at the top. And this is just one example of segregation mechanism. Another mechanism that can cause segregation is free fall. Imagine that you have particles of different size, density, different characteristics. If they're made to fall into an empty container, so when they will go down, they're going to displace the air. So as soon as this air will be displaced and the place of the air will be taken by the particles, that displaced air is going to go towards the side, and that is going to result in the particles based on their characteristics, especially their mass, to be uh, pushed to the side, and depending upon how much that push can be exerted on the particles, they're going to segregate. And as you can see here at the bottom, that the larger particles are affected less by this free fall segregation, whereas the smaller particles, the finer particles, they are affected more by the displacement of air, and they will have a higher density towards the edges, and the larger particles will have a higher density towards the center. So now it's obvious that segregation is a reality in the pharmaceutical setting, and it is a very important consequence that needs to be addressed. So there are several methods to study segregation in a pharmaceutical setting, and I'm going to take you through some of the current methods that are being employed in the pharmaceutical setting in order to study segregation. So one method that is already used to study segregation risk at GSK, for example, is the vibration-induced segregation study. So what happens in this method is that particles are made to free fall from a certain height into a chamber. Because the particles are falling down from the height from a certain height into this chamber, and this chamber, as you can see, is sledging downwards, or it has a slope. So the fall of the particles are going to result in segregation, and they're going to settle at different height. Uh, the particles are going to settle at different height on this slope. After the particles have settled, NIR, which is a method to detect the blend composition, is done by taking samples of this particle at different regions, and the NIR is going to tell the segregation intensity or the segregation risk of the particles in this chamber. So that is one method to study segregation. Another important method to study segregation risk at GSK is fluidization induced segregation study. So in this, what happens is, as you can see in this video too, that uh, air currents are actively induced from the bottom, and the air and that airflow is going to result in particles based on their characteristics, based on their mass, based on their surface area, and other characteristics. The particles are going to respond differently to the flowing air. Some particles, they will move towards the top, some of them they will settle to the bottom, and that is going to result in segregation of the blend, and that can be used to assess the segregation risk of a pharmaceutical blend. So this is another method. The third method, which is currently being used in UK, is the Wolfson tester. So what happens here is, it's very similar to the free fall method. What happens is, as you can see over here, that the blend is put in this uh, little silver plate on the top, and it is made to undergo a free fall through this sieve or through this tube or through this column, and the free fall is going to induce segregation in the particles, and then the particles are collected at different heights in this container at the bottom, and this and their composition is analyzed at different heights using HPLC, and the segregation risk is thereby studied. So these are the three methods which are currently being used at GSK and in other pharmaceutical companies to study the segregation risk. Now I'm going to talk about the method that I have worked on, or the method that I have developed to study the segregation risk of a pharmaceutical blend. So coming first to the objective of my presentation, or to the objective of my research, which was if the Quantos automation system can be used to evaluate the segregation risk of a pharmaceutical blend. So we wanted to see if Quantos, which is, I'm going to talk more about the Quantos automation system, what it is, and how we use that machine to study or evaluate the segregation risk in a bit. But the question that was posed was, if this system, if this Quantos QS30 system, or which is basically an automation toolkit, if it can be used to study the segregation risk of a blend. So basically, what is Quantos? Quantos QS30 is a machine, is an automatic powder dispensing system which is made by the Mettler Toledo, which is the company that is making which made this machine. So the primary use of this machine is for powder dispensing. It can dispense powders to a very high accuracy, and the mechanism that it uses in order to dispense powder is tapping. So you attach this dosing head over it, and I'm going to talk about all of this in detail in a bit. So you attach the dosing head, and the machine taps, and the result is the dispensing or the expulsion of powder from the bottom of this dosing head. So the aim of my study was to see if this particular machine can be used to evaluate the segregation risk of a pharmaceutical blend. So here you might pose a question that I just told you about three methods to study segregation which are currently being used or which are currently being actively used: the fluidization, the free fall, and the vibration method. So if we already have those three methods, why do we need to use this new method, or why do we need to develop this new method using the QS30 automation system? So in order to answer that question, we need to compare the way these two machines work. The first thing is that the automation system that I have worked on, it will require only 8 gram of powder for each analysis, whereas the fluidization, free fall, and vibration mechanism that I talked about earlier, they can need from 150 to 300 grams of sample depending on the type of method that we are using. For example, fluidization requires upwards of 300, whereas the free fall requires around 150 grams of powder. So as you know that APIs, they can be very expensive in a pharmaceutical settings, and sometimes they're very limited as well. So if we can develop this new method to study segregation, it can save us a lot of API. Number two, the QS automation system is fast and automatic. You just have to put the powder in the dosing head, put the vials on this chamber on this belt at the bottom, and leave it. Everything else from data accumulation, from analysis, from dispensing, everything is going to be done by the powder itself. And after the dispense of the powder, all the remaining steps that we have developed are also automatic, which also reduces the chances of error. It reduces it's much more quicker, it's much more faster, whereas the other methods, they're slow and manual, so they require much more input of human labor, and this the new system that I have developed can save a lot of manpower. Also, this machine is primarily a powder dispensing system, whereas the equipments that are used for the remaining three studies are special equipments that are acquired just to study the segregation risk. So this means that using this new system, uh, we will be able to take multiple use of the machine. So a machine which was primarily acquired for some other reason is now used to study segregation as well. So there's a multi-purpose of the machine. Also, it's very easy to use and it is safe because the powder is trapped inside this dosing head, so you can go and use up to dangerous compounds like OHC4 and OHC, and sometimes even OHC5. Whereas these machines, since the powder is direct in touch with the parts of the machine, you cannot use dangerous compounds. Also, this machine has a very small footprint, whereas these other machines, they are much larger in size. They require special rooms, and the availability of instruments is also a question because sometimes these equipments are not available, and you have to specially rent them out or acquire them. So there are a lot of troubles using the other traditional methods, and therefore we decided to explore in order to find solution to all these problems and to take advantage of the automation system. We decided to explore the new method of using the Quantos QS30 automation system. So now, how will we use this machine to study the segregation mechanism? So in order to do that, we designed the segregation workflow. So here's the blend segregation analysis workflow. So the step number one. So first of all, I just want you to focus on the first step. The first step is that we are going to fill in the dosing head. The dosing head is the small, you can see over here, is a small head that is attached to the machine. We'll fill this with 8 to 20 gram of unsegregated blend, uh, and then we're going to attach it to the machine. So talking more about the dosing head first, if you look at it over here, that this is how the dosing head looks like. This is the side picture of the dosing head, and this is how the dosing head looks from the front. Here you can see that there is like a small point. This is where it is going to be attached to the machine, and this is where the tapping is going to occur. And when it is going to tap, when the machine is going to tap over here or over here, it is going to result in this. You can see that there is like this pin over here that protrudes all the way from this head down to the uh, down to the bottom of this dosing head. So the result of tapping is that this pin is going to be expelled from the dosing head over here at the bottom, and that is going to result in the creation of a small hole at the bottom, and the result will be the expulsion of powder from the head. So this pin, not only does it taps, but it also rotates. The machine causes it to rotate and to tap simultaneously. And the openings, the dosing head has opening size which can be 2.5 millimeter or 4 millimeter depending on the dosing head you select. So there are many different kinds of dosing heads. Okay, so the second step of our workflow is that we are going to attach the dosing head to the machine, to the Quantos powder dispensing machine, basically the objective of my study today. So talking more about it, if you look at this video over here, you can see how this works. So you can see the dosing head rotating, and the powder is being expelled from here. So this dosing head is basically attached to the machine right over here, as you can see. And the machine does one thing, that it taps and it rotates the point over here, as I mentioned previously, and it results in the expulsion of powder from the dosing head into the vial below. So the third step is that now that we have dispensed the compound, or we have dispensed the blend into the dosing head. So the next step is we want to dissolve the API from the dosing head, from the powder. How we do that is that we are going to use a solvent that dissolves the API. So we will use that we will add that solvent into the vial, and then we are going to uh, cap the vial, and we are going to allow it to stir at magnetic stirring on a CM3. The purpose of this is to make sure that the API in the blend dissolves properly in the solvent. Since the blend together with the API, it also contains other excipients, we have to make sure that the API and the dissolved API in the solvent and the undissolved excipients are separated. In order to do that, we will filter uh, we will filter the solvent, and we will filter it using a centrifuge at 3500 rpm for 5 minutes, and the centrifugation is going to result in the separation of the undissolved excipients and the dissolved API in the solvent. Now the API has passed through the filter, is the filtrate, and the last step is that we want to quantify the amount of API in the solvent, and doing that will tell us what was the dispensed API quantity. Lastly, we are going to do some statistical test, especially the RST, which is going to help us determine the amount of segregation. And what RST basically does is that we are going to take standard deviation divided by mean in order to homogenize the samples with respect to their mean, and multiply it by 100. So let's talk about the statistical methods. Uh, the results that we are going to get are subject to the statistical method that we are using. There are many statistical methods that can be used: mean, standard deviation, absolute difference, coefficient correlation, or even a combination of these. But for our study, we have used RST, or coefficient of variation. The CV of a population, as I mentioned earlier, is standard deviation divided by mean multiplied by 100 multiplied by 100 in order to convert it into a percentage. So now I have walked you through the blend segregation workflow. Now let's talk about the experimental design and the results of my study today. So experimental design. So before jumping on to doing some experiments, we need to show that this method is a valid method. So we have to, in order to do that, we have to do some proof of principle studies. So the way I designed this experiment was that in the beginning, we have to prove that this method is a valid method. In order to do that, we wanted to see the segregation ranking between two different APIs, for which we selected compound A, which was 10%, uh, and compound B, which was 10% in its blend. So the API amount in compound A was 10%, and the remaining 90% were different kinds of excipients. And same is true for compound B, which is a different compound, a different API, which was again 10%, and um, the remaining 90% were the excipients. So we wanted to see the segregation ranking between compound A and compound B, and this will basically be used to we will basically use it to see whether our study is whether our study is giving us results that we want, and this will be uh, used as a proof of principle that our method is valid. So now we want to compare compound A, which is 10%, with compound B, which is again 10%. So here is the blend composition of the compound A or B. So everything in compound A and B is same except for the fact that one is compound A and one is compound B. All the excipients have the same composition, same size, and everything else is the same. But the thing is, if you see over here that compound A has a much larger particle size, it has a 343 micrometer particle size, whereas compound B has a smaller particle size of 39.8 micrometer. So we wanted to compare how compound A and compound B are going to show segregation results with our newly developed system. The reason we can use compound A and compound B is because from continuous manufacturing, we know that compound A has a higher segregation risk than compound B. So this is the result from continuous manufacturing, where something called acceptance value, AV value, which is a statistical test for which you take a difference in the mean and you add a constant K and multiply it by standard deviation. This is the statistical test that is used at continuous manufacturing. So for the API A, the compound A 10%, AP the AV value is between 15.25 and 46.41, and for compound B it's 5.23. So the higher AV value at continuous manufacturing indicates a higher segregation. What I mean to say by that is that compound A is segregating more than compound B. So if our method is valid, so the method that we have developed, if it can be worked, it has to reproduce the same result. It has to show us that compound A is segregating more than compound B. So we took this to our method, but before doing that we have to decide what kind of dosing head to use. So when I introduced you about the dosing head, I told you that there were two kinds of dosing head. One was 2.5 millimeter dosing head and one was the 4.0 millimeter dosing head. So first we have to decide which dosing head to use. So we took 50 milligram of compound A and we put it in a 4 millimeter dosing head and we put it in a 2.5 millimeter dosing head, and we dispensed around 120 between 120 and 140 samples from the 4 millimeter dosing head, which is the blue one, and from the 2.5 millimeter dosing head. Since we have 10% of compound A in a 50 milligram blend, we are expecting to see somewhere around 5 milligram of dispense. But here for the green one, you can see that the dispense is lower throughout, whereas for the blue one, we are seeing a much better dispense. So from this study, we decided to stick to the 4 millimeter dosing head instead of using 2.5 millimeter instead of using a 2.5 millimeter dosing head, because what we were thinking is that the 2.5 millimeter dosing head is causing some kind of filtration along with segregation, and we don't want a competing mechanism to come in between Therefore, we decided to stick to the larger dosing head size, which is 4 millimeter. So now coming back to the proof of principle. Remember that we had compound A and compound B. We know from continuous manufacturing that compound A shows more segregation than compound B. And compound A also has a larger particle size than compound B. So we are expecting to see the same trend or the same result from the method that we have developed. So here, when we did the test using our machine, the Quantos system, and using a 2.5 millimeter dosing head, this is what we observed. For compound A, you can see the segregation over here, and for compound B, which is the yellow, you can see segregation over here. You can visually tell by looking at the blue and the yellow that the blue one is more dispersed, or the blue one is more segregated than the yellow one. And remember, from the continuous manufacturing, we were looking at the same thing, that the compound A has a more dispersion or more segregation than compound B. Now we subjected this to our statistical test, which was the relative standard deviation, which is standard deviation divided by mean multiplied by 100. And when we did it for our when we did the statistical test, this is what we saw. We saw that compound A is showing a much more or a much higher segregation than compound B. So the results that we obtained were the same results, or the conclusion that we got from our machine was the same that we were getting from the continuous manufacturing. And this basically validates our automation system, that yes, our automation system is, or it can be used to study the segregation risk of a pharmaceutical blend. So after doing the first proof of principle, we jumped on to the second proof of principle study. And for this, for this proof of principle study, we are going to use the same API with different blend compositions. Remember, in the first proof of principle study, we were using two totally different APIs. But in this case, we are going to use the same API, but we are going to change the composition of the blend, or the characteristics of the API. So we are going to compare compound A 10% versus compound A 15%, and then we are going to compare compound B 10% versus compound B 5%. So same API but with different blend compositions. So now first we jumped on to, let's talk about compound A. We used two blends. One was compound A 10% and one was compound A 15%. Theoretically, we know that higher the blend composition, lower the segregation risk. This is something that we know from the theory. What I mean to say by that is that compound A 15%, since it has 15% of the API in the blend, it should show lower segregation risk than compound A 10%, because it is 10% API in the blend. From the continuous manufacturing, we get the same result. For compound A 15%, for the 15% of compound A, we see an AV value of 11.82, but for compound A, we see it between 15.25 and 46.41, as I previously showed you. So if our method is valid, again, it should show us that compound A is segregating less than compound A 15% is segregating less than compound A 10%. So when we did our study, this is what we got. The blue line is for compound A 10% and the gray line is for compound A 15%. And this is what we got after the RSD test. So the RSD of compound A 15% is 16.4%, whereas that of compound A 10% is 22.3%. This is exactly what we were expecting from the continuous manufacturing, from the AV value at continuous manufacturing, and from the theoretical framework, and we got exactly the same results. As you can see, that compound A 15% segregates less because it is showing an RSD of 16.4%, whereas compound A 10% segregates more because it is showing an RSD of 22.3%. So this again validates our method. So we did a proof of principle study 2B, the same thing as before, but we changed the compound. From compound A, we jumped onto compound B, which is a different kind of an API, and we have compound B 10% and compound B 5%. Here again, we will expect compound B 10% to show less segregation than compound B 5%. Because remember, the lower the blend API amount or the percentage of API in the blend, the higher the segregation risk from a theoretical framework. And from the acceptance value as well, we see that compound B um, 5% has an AV value from 3.65 up to 7.8, whereas compound B 10% has an AV value of 5.23, which means that compound B can segregate more than compound B 5% can segregate more than compound B 10%. So when we did our study and we subjected it to RST, so before going to that, look at the graphs. For the yellow one, you can see that uh, the segregation is over here, and for the 5%, here is the segregation. So the reason they are separated is because this is a 10% blend, the yellow one is a 10% blend, so it should be so the targeted value should be 5, and this is where it is going. And for this one, the targeted value should be 2.5 because it's a 5% blend in a 50 milligram blend. So when we did the RST, exactly what we saw. For the 10%, we see less segregation than the 5% because for the 10% blend, the RSD is 3.2%, which means less segregation than the 5% blend, which has a 3.7% RSD. So again, all these three things, the segregation ranking between two APIs as well as the segregation ranking between same API with different blend compositions, both from compound A and compound B, all these three experiments basically validates our method, and now we know that our method can be used to study the segregation risk. Now after doing the proof of principle studies, we jumped onto our first case study, for which we used a compound C, which was a very important pharmaceutical blend that was going to go to continuous manufacturing very soon. And there were three different compositions of compound C that were proposed. One was in which the compound C is 12.67% with the mean particle size of 37 micrometer, and the other was compound C 12.67% with the mean particle size of 22 micrometer, and then compound C 4.01% API with the mean particle size of 22 micrometer. So talking more about this, as I said that there are three different blends of the compound C. The blend 1 has the largest particle size and the highest API concentration in the blend. And compound 3 has the smallest particle size and the smallest API percentage in the blend of only 4.01%. From a theoretical perspective, we expect that the higher the API percentage, the lower the segregation risk, number one. Another thing that we know from a theoretical perspective is that the larger the particle size, the lower the segregation risk. So decreasing the particle size and decreasing the API concentration in the blend increases the segregation risk. So from a theoretical perspective, we are expecting blend 1 to have the lowest segregation risk because of the large particle size and higher API percentage, and blend 3 to have the highest segregation risk because of the smallest particle size and smallest API percentage in the blend of only 4.01%. So we did our experiments and we exactly saw what we were theoretically expecting. We saw that blend 1 has an RSD value of only 5.5% and is showing the least segregation risk, and blend 3 with the smallest particle size and the lowest API percentage in the blend is showing the highest segregation risk of 10.3%. And here in the graph, the results are displayed. So again, what we were theoretically expecting, we confirmed it through the experiment, and these were very good results and were a very happy news for us. And we recommended to the team to use the blend 1 as their preferred blend composition in order to make the in order to make the final pharmaceutical blend. So we, after doing the first case study, we decided to do another case study in which we wanted to see the impact of excipient particle size distribution on the segregation of API. So in the first case study, remember, we were changing the characteristics of the API and seeing how it affects the segregation risk of API. Here in the second study, we are going to change the blend composition or change the characteristics of the excipient. And we're going to see how changing the excipient affects the segregation risk of the API. So the API is going to be the same. So the API that we are going to use in this case is basically compound B, and all the remaining are the excipients, and the excipient that we are going to change is the excipient 2A, which is the major, which has a very high percentage in the blend of 56.4%, which means that over half of the blend is made up of excipient 2A. So what we are basically doing is that we are changing the characteristics of excipient 2A. We are basically changing the particle size distribution of excipient 2A, and we are going to see how changing this affects the segregation risk of compound B or the API. Coming back to the particle size distribution, for excipient 2A, the mean particle size is 106, whereas the compound B is only 39.8 micrometer in particle size. So basically, you can see from here the compound B is smaller than excipient 2A. So we basically decided to change the size of excipient 2A, and we used these three different sizes. For 1A, the particle size of the excipient is going to be 50 micrometer. The 2A, which is the standard one that we previously used as well, is around 100. And for 3A, it is 180. So we will change the excipient particle size, and we will see how changing it basically affects so we will see how changing this affects the excipient, how excipient changing affects the API segregation risk of compound B in our case. So when we did this experiment, we were not able to see a very clear trend. What we observed was that for excipient 2A, when the particle size is 100, the RSD value is 3.2%. When we decrease the excipient 2A particle size to 50 or we increase it to 180, both the cases the RSD value increases or the segregation risk increases. So there is not a very clear trend over here, but in order to verify this trend, we need to do more experiments. It will be too early to draw a conclusion. But some important things to note from this experiment: if you look at the orange line, you will see excipient 1A, which are the largest particle size, or 180 micrometer. So you will see that in the beginning the segregation is low, but after somewhere around 50 to 60 dispenses, the segregation risk increases a lot, whereas this trend is not seen in the other two excipient sizes. So more studies needs to be done in order to draw some solid conclusion, because we cannot see a clear trend. But nevertheless, this was an important case study. So lastly, we did a case study of compound D. This is a compound all by itself, and we wanted to see what is the segregation risk of this compound. This compound, basically the compound D, is a 50%, it has 50% API in the blend. And we wanted to see how it is going to affect, what is the segregation potential or the segregation risk of it. From the continuous manufacturing, we already know that it experiences very high segregation. But our value was only 4.4%. However, what does this RST of 4.4% means? Does it mean that there is low segregation, or does it mean high segregation? The answer is we don't know. We cannot go back and compare this RST value with the other RSTs that we got from blends that were 10% or 12% or 5% and say that since their RSD is 4.4% and shows less segregation. This also means less segregation. We cannot do that. The reason being because this is a blend uh, with a 50% API, and changing the API basically results in a change in the segregation risk. So if you want to compare this with the studies that we have already done, then you need to bring down uh, the API composition of this blend from 50% to the level uh, to the compound that you're comparing to. So this study basically can be used to is a good study to make relative comparisons, but it cannot be used to um, look at a compound, look at its RSD value, and predict how much segregation is going to happen at continuous manufacturing. Uh, or at least, or we have to find the right statistical test which can be used to do that. So basically, the outcomes of the project. So this project can be used to compare segregation ranking between different APIs which have the same blend composition, like the one that we did for blend A when we use the 10% and 15% of the blend. Um, It can be used to see how changing the API particle size and composition affects the segregation risk, like we did in the case of compound C. And it can be used to explore the effect of changing excipient size on API segregation, as we did in the case of compound B, where we use the excipient 2A, 1A, and 3A. However, the limitation is that it does not exactly simulate all the segregation mechanisms that are happening during the continuous manufacturing. We don't know what are the exact mechanisms that happen at that stage, and so uh, we cannot say with the sure shot that this machine is replicating the segregation mechanism that happens in the continuous manufacturing. Moreover, on top of that, uh, the because we have not been able to find the right statistical tool, this study is very useful for relative comparisons between between different APIs, or we can study how changing blend composition or excipient size composition is going to affect segregation, but we cannot take out a compound, find its RST, and predict the segregation that is going to occur at continuous manufacturing. However, we can say that between, say, compound A and compound B, compound A is going to segregate more and compound B is going to segregate less. So for this project, I would like to thank Kenneth Wells, our scientific director, who gave me the chance to work uh, here as a co-op for the high throughput automation lab. I would like to thank Eva Wu, who is my advisor for this project, and all the other team members, including Christopher Nunn, John Kina, Katie Grenias, Matthew Bahar, Sophia Koshevili, Sharon Metromes, James Thomas, Summer Stephens, Brendan Turner. I would like to thank all of them for their continuous support, for their continuous help, without which this project would not have been possible. And from PPE, I would like to thank Parin Desai, who was also my advisor for this project. And along with him, I would like to thank Sharias, Cameron, Richard, and Kurt. All of you really helped me to solve this project. Thank you so much for all the help. Thank you.